This is an example of how vilma works with GBIF data to map evolutionary diversity. Usually, researchers and governments use species richness to compare areas and guide conservation decisions. However, areas with similar richness can be very different in their evolutionary histories. Then, phylogenetic diversity indices help to capture this hidden dimension of biodiversity. While these indices are relatively easy to calculate at local levels, they become more challenging at large spatial scales. This is where vilma becomes useful. Using occurrence data and phylogenetic tree, it calculates multiple phylogenetic diversity indices and returns spatial outputs ready to visualize, export, and analyze. It also includes a shiny app for users with less programming experience. Because vilma was mainly built for work with occurrence data, the package offers the opportunity to transform the vast amount of information available to GBIF into maps of richness, alpha phylogenetic diversity, null models, beta phylogenetic diversity, and even phylogenetic regionalization. To use vilma, you only need occurrence record and a phylogenetic tree. You can go to the GBIF website, find the data for the group of interest, and download it. Then you can go to BirdLife website and retrieve a phylogenetic tree for the same group. Because vilma requires the input CBS file to have 3 columns, species, longitude, and latitude, you can open the download occurrence file, extract those columns into a new CBS file, and save it. With just a few clicks, you're ready to start analysis. For this example, I will use the Shiny interface. To open it, first install the package using the app tools, then you just need to set the working directory, load the library, and run this function, which launches the app. Immediately the app will open in your default browser with the welcome page. You can click continue and the about vilma page will be shown. Click continue again and you will see the how to use manual. This page contains a short guide to the package and the tools available in the app. You can scroll to the end and click next to start analysis. Now in this page, because vilma works with rasters, you will convert the occurrence points into the raster object. Just click Browse, open your CVS file with the 3 required columns, select the projection and the resolution of your raster, and run the analysis. When it's done, you will see the results as an interactive map where you can zoom in out, select between the 2 available layers, richness and abundance, and also check the summary output, which shows information such as the number of species, records, and cells. Now you can move to the next page. On this page, you will calculate the alpha phylogenetic index. First, you load the phylogeny, select one of the available indices, and configure its parameters. Now that everything is set up, click the Run button to view the interactive map and the summary output. The interactive map, you can navigate through the 3 different layers, richness, abundance, and the index PD values. Then you can just continue to the next page. On the new model page, you can test whether the observed phylogenetic patterns differ from random expectations. vilma offers 2 options, a global analysis for the whole studio area or a cell-based analysis where the statistics are calculated for each pixel. You can select the number of iterations and the resample methods such as taxa, range, neighbor, or regional. Global analysis returns a histogram of observed and null PD values, while cell-based analysis returns an interactive and standardized effect size map and a summary statistics. Depending on the study area and method, this step can take several minutes. On the beta phylogenetic diversity page, you select the beta indices and set its parameters and run the analysis. The results are shown as an interactive map and summary window. vilma calculates the average index value per cell and also provides ordination outputs using PCOA and MDNS axes. Finally, on the last page, you can export all the results from the alpha, beta, and null models analysis. vilma saves the available rasters, tables, and log files, and can also generate an R script with the functions used during the session, making the workflow reproducible. So, as you already saw, with 2 files, GBIF occurrence and a phylogeny, you can obtain different spatial files with a lot of evolutionary information that you can continue using in research, planning actions, conservation, and other applications. That's the main idea of vilma. To use occurrence data and transform them into spatial evolutionary information in an easier and more accessible way. Of course, for further information, you can visit the vilma repository in GitHub, but I strongly recommend visiting the website. There you will find many tutorials and detailed explanations of everything, the functions, the ideas, the formats, the outputs, and also every tutorial starts from scratch, including how to retrieve the data from GBIF or BirdLife, how to use the files, how to transform them, and how to run the analysis.